shall we? All right. So my name is Mahmoud Moradi. I'm an assistant professor at the Chemistry and Biochemistry Department, University of Arkansas. And I'm going to talk about complex transition pathways and how we can explore them. So many of the elements of this talk have been already taught in the previous sessions by Chris and Jerome and others. And I will not focus too much on the details of individual methods. Instead, the approach that I have for this talk is more how we can actually combine them, how we can actually study a realistic system, rather than talking about the details. Although I will talk about some of the details, but rather than talking about the details of what umbrella sampling is, for instance, or how we can combine it with replica exchange, I will talk about how we can use things like non-equilibrium simulations, like SMD type simulations, string method with storms of trajectories, umbrella sampling with replica exchange, and analysis methods, and things like that. How we can combine them to study a realistic system. The idea basically is you probably need to combine these methods if you want to study a realistic system. Why? Because realistic biomolecular systems, especially if you are interested in something like the structural biology of proteins, like the conformational changes, similar to what you see here in this schematic picture, which is one particular type of channel called the mechanosensitive channel of large conductance, one of the systems that we work on in the lab. So if you are interested in this kind of transitions, this kind of complex conformational changes, you shouldn't actually expect to have a code that you can sit there and just push a button and does the job for you. Because it's complex, it's complicated, it requires knowledge. You do need to have some knowledge. There is no mathematical solution, as far as I believe, at least, for a problem like this. If you want to mathematically solve the problem of protein dynamics, it's not going to work. It's just going to be an impossible problem. Therefore, you do need to input information. Like one of the first things that you need is you do need to have a crystal structure. So if you don't have a crystal structure, you better not to study it. If you are interested in this kind of things, if you're interested in other things, that's fine. But if you're interested in studying the detailed conformational changes of a protein, well, you need to start from a crystal structure, something reliable. But that's not enough, because crystal structures usually capture proteins in one state or maybe multiple states, but not all states. And interestingly, most of the time they capture them in the inactive state. Like for channels, a lot of channels are crystallized in their inactive state. Because in the crystallographic conditions, you shouldn't expect your protein to actually function. It would be probably in an inactive state. Like in this particular case, this channel of the MSCL channel here, this is the crystal structure. The channel is closed. That's what we actually do expect. So suppose that you want to do something with this protein now. You want to design some drug for it, an inhibitor for it, for instance. Well, you have it in the inactive state. You probably want to know how it functions, how it actually works, before you want to design something for it to inhibit it. So the idea here is larger scale conformational changes are important because they exist and they play a very important role in the function of many proteins. And to study them, we need to have a lot of experimental data combined with a lot of computational techniques using also probably big supercomputers. So again, someday you can do the things that I'm going to talk about in a regular computer, but right now, most of the stuff that I'm going to talk about do really require large supercomputers to be able to do. Maybe the initial parts are easier, but then as we approach to the more promising parts, where we're actually going to do things that are going to give you reliable information about the structural transitions of proteins, at that point, right now there is no easy solution. You have to use large supercomputers, you have to use relatively complex methods, combine them, and so on. So I hope it wasn't depressing too much, but I have to just tell you the reality. But it's not impossible, it's possible, and you can actually study transitions like this. And this is actually a new thing, I would say. It's something that is only possible, has become possible in the last few years. And you can find different independent research groups that are doing things like this in a reliable manner. I mean, in the old time, people did things that don't really make a lot of sense. Like, for instance, you want to study the transport process. They would just take the substrate and pull it out and from the protein. And that they would expect to get some sort of pathway or some sort of free energy. That doesn't really work because the binding and unbinding, especially if it's like a channel or you have a particular binding site which requires rearrangement of the side chains and the rearrangement of the helices. That kind of problem is not something easy to understand. You require probably multiple collective variables. You require multiple sampling techniques to be able to attack such a problem. So in the old time, people did try ideas that were not really sound. These days, at least some research labs are trying more reasonable methods. And I will talk about some of these ideas. So there is a tutorial associated with the talk that I give, which is available. And you can do it either today or whenever you go home. The tutorial is accessible to you on the website, ks.uiuc.edu. Tutorials. Yeah, so yeah.
Training tutorials.
Yeah, training tutorials. The address. So you can actually access the tutorial and go through it. So the tutorial doesn't cover everything that I talk about today, but it does cover a big part of it. For the other parts that you cannot find in the tutorial, you can contact me and I will provide you with the codes until I actually put it together in the form of another tutorial. Okay, so what I'm going to talk about today, we have two sessions, is outlined here. This covers the two talks that I have this morning. So first, I will talk more about why we need complex methods and why just equilibrium simulations don't work. Even for qualitative understanding, they don't work. So forget about the quantitative understanding of such problems. Forget about free energy calculations. even if you want to understand things qualitatively, equilibrium simulations don't work for larger scale conformational transitions, and I will actually tell you why. Then I will discuss the methodology, and I will particularly talk about two ideas that weren't discussed before in the previous sessions. The first idea is how we can actually decide what collective variables that we should use for our studies. That's one question I try to provide you with one way of figuring out. And that's an empirical approach. So it doesn't tell you what collective variable to use. It tells you that if you do have multiple candidates, which one to choose. The second idea is about the combination of free energy calculation methods and string method with sorts of trajectories or other pathfinding algorithms. So rather than doing free energy calculations in a generic way in a space of a collective variable, you can actually do free energy calculations along transition pathways that have been optimized using some transition path optimization method. And that transition path optimization method could be itself using another pathway that is generated using non-equilibrium simulations that basically is along a particular collective variable or has been generated using a particular collective variable that you have optimized using the first idea. So really this talk is about how to combine these ideas, how to combine non-equilibrium simulations with path optimization, with free energy calculation methods, and so on, and how to iterate, how we can actually do this in an iterative manner. Like sometimes you do free energy calculations and your results don't make sense. It happens a lot. It happens more than you can actually imagine. What to do at that point? Do you want to just publish it? Do you want to just put it aside and start another project? So I will actually try to tell you what to do at that point. And there is a way of using that data that you know is not reliable to be able to start another set of simulations that might end up being reliable. And that is the iterative part. So how you can use data which you know is not good, samples that are not necessarily good samples. They haven't sampled the space that you're interested in a very good manner, but they are samples, and it's you spent your allocations, your computer allocations on them. So you have all these samples. What do you want to do with them? So I will talk about that a little bit. And if you have time, so it all depends how it goes, but if you have time, then I will talk about a particular idea which is not in the tutorial. Hopefully it will be added at some point, but that is kind of revisiting everything that I'm going to talk about and making it more accurate using a theoretical idea, which is a very general theoretical idea about the free energy calculation methods and transition sampling and things like that, which is basically going beyond the Euclidean geometry that we usually use and start using Riemannian geometry, which I argue is the more reasonable approach to collective variable space. Okay, so I'm going to start with basically an example of what I'm talking about when I say larger scale conformational changes. So most of you probably have a very good idea what it means, but just I give an example related to the kind of research that I personally do in my lab does. So that is actually the larger scale conformational changes in membrane transport proteins. So typically you have two types of membrane transport proteins. Some of them are transporters and some of them are channels. In both of them, you usually do see larger scale conformational changes. For transporters, it's more common. Almost all transporters undergo some sort of larger scale conformational change. And that is because since they are usually active, they transport their substrates, they transport the molecules against their concentration gradient. They have to undergo an alternating access mechanism, which basically says the transporter cannot be, or the binding site of the transporter cannot be accessible to both sides of the membrane at the same time. So they have to either be in an outward facing or an inward facing state. And this requires larger scale conformational changes. So the protein has to change its shape to be able to do something like this. Channels, sometimes it's not really a very large scale conformational change, but also you do see that in a lot of channels that have to undergo conformational changes. And examples of it is what, one of the examples is what I showed you in my first slide, the mechanosensitive channel of large conductance that opens really a lot, and it requires a very large scale conformational change. So in channels, it's also common to have large scale conformational changes. In many other proteins, it's common to have large scale conformational changes. And usually these large scale conformational changes are necessary for the function of the protein. So if you are interested in the function and a structure relationship, then this becomes basically the main question to answer: how this protein undergoes large scale conformational changes and how it's coupled to chemical events. How, in this case, for instance, the transport and the largest conformational change of the protein is coupled to the binding, transport, and unbinding of the substrate. And if there are multiple substrates involved, then it becomes more complicated. There is a coupling between all of them. You have one sodium, one chloride coming in, binding, and then there is also a proton involved, and it becomes complicated. But you do need these chemical events to trigger those larger scale conformational changes, and this is something that basically can be called the chemomechanical couplings in proteins, which then requires, again, understanding the larger scale conformational changes. So why equilibrium simulations are not good for this kind of studies? Well, I have actually a kind of a particular case study here that I can show you. So this is a membrane transporter. It belongs to the proton-coupled oligopeptide transporters. Basically, in humans, it basically uptakes dipeptides and tripeptides, and is coupled to proton flow. That's the source of energy for it. So it's a very important protein, and we would like to see how it works. We would like to see how the proton flow or proton binding and unbinding is coupled to the larger scale conformational change of this protein, and how that is coupled to the transport of the peptide. Now, the chemical events that we are interested in include binding of a proton to the binding site, binding of a substrate. And one thing we know about this protein is it's actually a promiscuous transporter, in the sense that many different dipeptides and tripeptides can be transported by this, although the affinity is not the same for all dipeptides and tripeptides. So we also want to understand the substrate specificity and promiscuity. So we try to model this protein with different dipeptides, 3 different dipeptides, and also with and without the proton. So we had 8 different conditions. And this is a typical thing. So we want to study the chemomechanical couplings, you try different conditions of your protein, or you put your protein in different conditions, and then you run a simulation. This is something that a lot of people do. This is like a very common approach that people do, and then they try to understand what happens if the proton is there or if the proton is not there. And I'm going to actually tell you if this is a good idea or not. You judge yourself. So what we did was we ran a 400 nanosecond simulation for each condition, and we repeated that twice. So basically, we had 2 repeats of the exact same simulation. So this is the part that some people don't do. They just do 1 simulation for every condition. Also, 400 nanosecond, you can do also, in our recent work, we've used 2,400 nanosecond, basically 2.4 microsecond for another transporter. And actually, the longer you go, you realize how worse your sampling is. So if you just do a very short sampling, you think that, oh, I observed this, this transition, or I observed this side chain is moving. But when you run this longer and you repeat this, you realize that you're not really sampling good. It's more random than anything. And this is what we will see. So this is 6.4 microseconds of simulation, which is a big amount. And let's see what we get out of it. So I'm going to show you 2 types of time series. One type is called the local conformational changes, things that are related to the local conformational changes, like the movement of the side chains, like salt bridge formation. You have 2 amino acid side chains, and they form a salt bridge. This is a local thing. They can easily do this, although it might be coupled to some larger scale conformational change. But it's basically, it involves two side chains, so it's a local thing. Another type of conformational changes are global conformational changes. Like in this case, the angle between the 2 bundles of the protein, the N bundle and C bundle, they form an angle. And this angle is important because the way that this protein functions, people call it the rocker switch mechanism. So it would involve something like a rocker switch movement, which means the angle between the 2 bundles changes. So if you characterize that angle, that tells you something about the larger scale conformational changes. And you can also look at other things, like many people don't look at angles, they just look at distances. So that's also another common mistake that some people do, like they think that they can express everything in terms of distances. So if they want to express something like this, rather than using the angle between the bundles, they find 2 loops, and then they look at the distance between the 2 loops, and that's it. That's how they measure the conformational change of the protein. I don't understand why some people think that for studying a complex system like this, they can resort to very simple measures like distances all the time. Like, just take a textbook and learn what an angle means. And maybe you can use the idea of angles instead of distances. Well, that's one way of characterizing the larger scale conformational changes, but it's problematic. For instance, the loops, they move all the time. So is it the larger scale conformational change that is happening, or it's just the loops that are moving locally? So it doesn't distinguish between the 2. So these are also other larger scale conformational changes, or measures that can capture larger scale conformational changes. The angles between individual helices, the interhelical angles. So that tells you more specifically whether the extracellular loop, extracellular gate, and intracellular gate are opening or closing. So we know, again, from the literature, from what people have suggested before, that these particular helices are involved in the opening and closing of the periplasmic loop and the cytoplasmic loop. And if we measure their angles, it tells us something about it. So we start with defining some measures. And by the way, I'm not just talking about this particular protein or running equilibrium simulations. These ideas are things that we should use in then our free energy calculations, in our non-equilibrium pooling simulations, and so on. So it all starts from here. If you want to study the protein, first you need to come up with some good measures of how to describe the conformational changes, both local conformational changes and global conformational changes. And you need to have a very good idea that which one is actually a local thing and which one is a global thing, because you have to treat them separately, and we'll talk about that. Okay, so the local conformational changes are easier to study using equilibrium simulations. You usually could see something meaningful and reasonable even with relatively short simulations. Like if you're protonating a particular residue, you may see something happening in a reproducible manner. So here I have the unprotonated protein, so that there is one particular amino acid, and that is the E310 that you can protonate or unprotonate or not protonate. And the unprotonated one in the apo, so no substrate, was repeated twice, set 1 and set 2. And in both cases, the salt bridge distance is basically telling you that this salt bridge is formed. However, the same system, when the E310 is protonated, the salt bridge isn't there anymore. And it makes sense. So you are protonating this glutamate, and it doesn't form a salt bridge anymore. Almost you don't even need to do simulations for it, but you can see it in the simulations. So this is another example when you do have the dialanine there. Again, when the system is unprotonated, you do have a salt bridge. When it's protonated, you don't. So this is the kind of things that you can do, and it makes sense. And these are some local conformational changes. Usually, it may tell you something if it's reproducible. And if you run long enough, it will tell you something, and you can use. But what about the global conformational changes? Here, I'm looking at two different things. One is the CN bundle interdomain angle, which is the angle between the two domains. And the other one is the loop-loop distance that I also showed in the previous slides. So in both cases, you see that when you have two different conditions, in one case it's the unprotonated apo, the other case is protonated with dialanine substrate. They look different. So they do have a different behavior. You have run 400 nanoseconds. And in one case, the blue one, which is the unprotonated apo, the angle is larger than when it's deprotonated with the dialanine peptide. So we have discovered something very important. Let's publish it. But let's actually just run it one more time before we publish. We run this one more time. There is no distinctive behavior. It's just the same thing. It was just completely random that one of them just had a larger angle in your single simulation that you ran. And this is very common. So just repeat your simulation one more time before you publish it. So when you repeat this, when you do this more rigorously, you realize that, well, there is no real distinction between different conditions, at least when you're looking at these two. But not everybody necessarily repeats the simulation before they publish. So this is the exact same system that was published in this PNAS paper a few years ago. This is the exact same simulations ran for 200 nanoseconds each. They see a different behavior, and they make a lot of claims about the structural biology of the system and how the structure is basically related to function and so on. We did this for multiple different global conformational changes and 8 different conditions to see whether we actually can observe even one single global conformational measure that does show a distinctive behavior in 2 different conditions. And we weren't able to find even one. So if you look at different measures, so these are 4 different global conformational measures in 8 different conditions. And the red and blue, one is the set 1, the other is the set 2. So it's the repeat of the same simulation, and you look at the average and the standard deviation. So they are all just random. You cannot find anything statistically meaningful that tells you that, well, when it's protonated, this is opening more. When it's unprotonated, it's opening less. So this is the problem with using equilibrium simulations to understand global conformational changes. And it's very easy to understand this. Like, think about global conformational change as a transition like this in the space, in the reaction coordinate space or your atomic position space. So when you're looking at the equilibrium simulation of one of the end states, let me just finish. You basically see some behavior which may not have anything to do with the actual global conformational change and the global transition, the state transition that you are interested in. It could be completely orthogonal to it. And it's just not possible to make statements about this transition based on simulations of the end states when the transition doesn't happen. If the transition doesn't happen, you can't make statements about it. So this is basically general take from it. So
when you say statistically insignificant, did you do a test of significance? Do you have a p-value for?
I do not remember if we did for that particular project or not, but we've done this for other projects, and it's very difficult to get really anything significant in terms of the global conformational changes from this kind of simulations, typically. So I'm not saying that it's impossible mathematically. Sometimes it might be possible, but usually you better have the transition before you want to make a statement about it. So this is the general idea.
Yeah, so I'm not directly involved in this specific field, but I'm sure you're aware that there's a few people who have made a career based on predicted conditions from fluctuations in one's end state.
Mm-hmm.
And I think, again, I don't know that literature very well, I think they have a few successes to show for their approaches. So what's your general take on that? Do you think those are flukes, or there's just a small category of cases where that would work?
No, I mean, I believe that it's possible that we have examples that it works, but it's not a general approach that can be taken seriously, in my opinion, because of this argument that I have here. So if you want to solve this problem in a systematic way, that how to study the larger scale conformational changes of the protein, I would not recommend that method, although it may work in certain situations. Yeah, and I have an example that it didn't work, and I showed this very explicitly that, well, it just doesn't work for this system, and probably for a lot of other systems. But, I don't have any mathematical proof that you always get it wrong.
Yeah. So it is possible for me to get lucky and try to use equilibrium and actually cross that little bridge?
Well, I mean, if you just use equilibrium simulations and you expect to cross the bridge, that's definitely possible depending on your system. So if it's a very slow process, then the chances are you won't. Like in this particular case that I showed you, this is a membrane transporters, and the time scale for membrane transporters function is usually several milliseconds minimum. So you don't have the chance of,
for that.
Yeah, so you won't be able to observe that transition if you just run a microsecond simulation. So it depends on the system that you're studying. So my focus here is on larger scale conformational changes that occur on time scales that are beyond the current molecular dynamics simulation schemes that we have. The equilibrium simulations cannot capture them. And membrane transporters are one example. So there has never been a complete transition observed for a state transition observed for a membrane transporter. You may actually end up with going from like open to occluded or occluded to open, but you won't go from inward facing to outward facing. That's just very, very slow. So there are examples of proteins that undergo transitions that are within the time scales that are accessible to regular molecular dynamics. But this talk is not about that. Okay. So now that I kind of made my argument against using just regular equilibrium simulations for the study of larger scale conformational changes, the question would be, so what to do now? The idea that I kind of promote here is to start with generating some pathway based on the knowledge that you have of the other state, kind of an empirical way of reconstructing the transition, and that involves using some experimental data about what the other state might look like. Sometimes you might have a crystal structure. Sometimes you might have a homologous protein with a structure. Sometimes you might have some data on the distances of some of the residues, like from Fred or deer or things like that. So try to actually generate the transition, and we will talk about how that can be done. And then try to make it more rigorous. And that would be try to optimize the pathway and try to quantify it using free energy calculation methods. So the first step in the strategy that I'm talking about here is usually using non-equilibrium pooling simulations, or SMD basically, but not necessarily using distance as your collective variable or RMSD as your collective variable. You can actually come up with different ideas for doing this. Like in this case, it's the orientation of these two helices, these two yellow helices that you see here, that we try to impose a conformational change on the protein and open it. And it comes from this rocker switch mechanism idea. There are some experimental evidence for it from the looking at different crystal structures of this family and some other data that proposes a rocker switch mechanism, saying that the protein kind of undergoes a somewhat rigid rocker switch movement, which is not true for all proteins or all transporters. This has been suggested for this particular family, the major facilitator superfamily of transporters. So we can try this idea and other ideas. And especially if you are going to study a larger scale conformational change, a complex conformational change, it's a good idea to try to use the collective variables that you have in mind in a simple SMD simulation before you want to do ABF or umbrella sampling or expensive calculations like that. Because maybe your collective variable is not good enough to even give you the transition. One possibility is that you may actually see the transition, but the end state may not be stable. In this case, we actually tested that. We took the final state, we equilibrated it. First, we actually put restraint on it for a little bit, and then we removed the restraint, and then we let it just run freely. This outward-facing state that comes out of this non-equilibrium simulation, that comes out of a very simple idea of the rocker switch mechanism, which then, in terms of collective variables, is translated into the movement of these two helices, just orientational change of these two helices that was imposed using the orientation quaternion collective variable. So once you freely run the simulation, you see that it's actually stable. It actually stays open. And it's also an outward-facing conformation. Why? Because if you, for instance, count the water molecules, in this case, you're looking at the number of water molecules along this z-axis. The red one is the initial equilibrium simulation of the crystal structure-based model, which is in the inward-facing state. And it shows an inward-facing state. So you have an opening, you have more water molecules here, and then nothing here. The outward-facing, the model that we generated using this approach, if you run that for a couple of 100 nanoseconds and then look at the behavior, first you need to see if it's stable or not, and then you need to see whether it actually has the function that you expect from it. Like in this case would be the outward-facing function. And when you look at the water molecules, you see that again, you'll see a lot of waters on the X and the periplasmic side, but not in the cytoplasmic side. You can also use other things like using hole to characterize the opening or any other approach that characterizes the functionally relevant feature of the system. In this case, the closest thing that we could come up was just counting the water molecules. Because if water can access the binding site, then the substrate can access the binding site as well. So this is the first test. So if you do have a collective variable, it should be good enough to be used in a non-equilibrium pooling SMD simulation and give you the transition that you're interested in. A lot of times it's not successful, especially if you use a simple one, like if you use a distance-based collective variable. In this case, I could have used some of these loops and moved them. Just define a distance between two loops on the extracellular or the periplasmic side, and then pushing them. That was a possibility. But unfortunately, that didn't work for this system. It didn't actually trigger the conformational change that I wanted. It just opened the loops. So the collective variables that you define should be good enough, at least to generate the transition that you're interested in. And it's not just the end state. The intermediate states are sometimes important as well. For instance, for other transporters, we've used some collective variables that have resulted in an intermediate which is open to both sides of the membrane. We know that's not possible for a transporter because it defies the alternating access mechanism. However, for instance, if you use a simple collective variable like RMSD, it may give you the final state, but then the intermediate state may not be right. And so it takes a lot of double checking for the consistency to make sure that you are using a good collective variable. And I will talk about that a little bit more. So I will talk about this empirical search for protocol optimization, which primarily involves in choosing the right collective variable for your simulations, for your enhanced sampling simulations. And I will talk about some empirical ideas for it. One I already talked about, which is qualitatively just check to see if it has given you what you expect from a transition. But there are also other factors that I will talk about. And after that, I will talk about combining the free energy calculations and pathfinding algorithms. So some basic ideas about sampling. For free energy calculations, you require some sort of dimensionality reduction. That's why people define collective variables, because it basically allows you to reduce the dimensionality of the system. and work in a simpler framework. You can also, rather than using ad hoc collective variables, you can use some more automatic methods to design collective variables, like use PCA, use diffusion maps, ISO maps, things like that. And some people are trying those ideas as well. What I talk about here is independent of how you come up with your collective variable. You may come up with it using some knowledge-based ad hoc method or using some automatic method, but whatever collective variable you are going to use should actually be good enough for enhanced sampling based on the ideas that I will talk about. If it's coming out of some sophisticated code, it doesn't mean that it's necessarily a good collective variable. It should still give you the transition that you want, and it should still it shouldn't require a lot of non-equilibrium work, as I will talk about. And another idea which is related to dimensionality reduction is reducing the sampling to a one-dimensional pathway. So that's what the pathfinding algorithms try to do. So you may end up with, you may not be able to end up with one single collective variable. You may end up with multiple collective variables, or sometimes you may even not use collective variables at all, but still you can reduce the dimensionality by finding a good one-dimensional pathway, basically, and sample around that pathway. So the idea of transition path sampling and optimizing the transition path is related to the idea of collective variables. They are both basically trying to reduce the dimensionality and make sampling easier. And the path is, of course, can be obtained from, for instance, the transition path algorithms. Yes?
Is there any experimental data that we can use to find our transition path or confine our search?
Yeah, absolutely. The first thing to do before you design your collective variables, or what you want to do, should be gather all experimental data available to you. All the crystal structures of your protein, all the crystal structures of the homologs of the protein, all the experimental data on the distances of certain residues through FRET or through double electron resonance spectroscopy. Anything that is basically useful to tell you something about the structure of the protein should be gathered and used either quantitatively or qualitatively. So, and collective variables allow you to do that. Collective variables can provide you with the framework to impose all those restraints. The very first example that I actually had in the slide, which was the MSCL channel, it was actually based on the model, the open model that we used for our transition path sampling and for transition path optimization and then free energy calculations, was based on something like 27 single molecule FRET measurements on MSCL channel that we put together, and we used that as the final state. Although it turned out that the final state that we observe in our simulations is not the same as the final state that the experimental data gives, because they are not really in the same conditions. We should always remember that all these crystal structures come from certain conditions that do not necessarily match physiological conditions and also your own simulation conditions. The same for other experimental methods. So in that case, for the mechanosensitive channel of large conductance, the way that they triggered this opening was not a physiologically relevant method. They did this by basically asymmetrically changing the membrane composition. So, which is not the so you wouldn't expect necessarily the same kind of opening. It was good enough for us to use as our target, but then later, during all these optimizations, we ended up with a pathway which gave us a different end state. You can always blame the force field for it too, but I wouldn't really go that path. Yeah.
Let me just interject with a little suggestion. If the starting knowledge you have is a bunch of structural models with different possible states or different states, one thing you could do now is load up all of those models in VMD and use the VMD GUI to play with variables and see what values they give in your models and what differences you can resolve or not resolve.
Absolutely, and I will actually talk about an example of it. I didn't necessarily use that GUI when I did it because that was a long time ago, but I did it manually, and we can look at an example of that. So, so these are some ideas, formulating these ideas. We need to define reaction coordinates, and that is basically a set of system-specific collective variables. And the GUI on VMD helps you a lot with that. So you can design different collective variables based on different values that you observe. So you can come up with a set of system-specific collective variables. Then you can search for efficient pooling protocols, or SMD protocols, basically. And that is combining these different collective variables, choosing the right simulation time. Sometimes 1 nanosecond might not be too long enough. The simulation time needs to be long enough, but you don't want to go too long because you don't want to basically spend too much computer time on it. The force constant that you use, this kind of parameter, the pathway that you can choose. If you have multiple collective variables, you want to do SMD, you can actually choose a particular pathway to go through. So you can play with these things. And you can actually do this empirically. You can actually do this, you use ideas based on what you see from the crystal structures, from other experimental data. You come up with what you think is good. And then you can see whether it makes sense or not. And then if you have multiple ideas, you can try them and see which one makes more sense. And then finally, once you do have a pathway, you can optimize that pathway and then calculate the free energy along that pathway. So make it basically quantitative. So this kind of scheme is something that I've been working on, different pieces of it, using the different approaches that are mostly already known. The umbrella sampling, or the adaptive biasing force, like adaptively biased molecular dynamics, metadynamics, SMD, string methods. So these are all known methods. So what has been more interesting to me has been combining them and making them work. So basically, the idea is different sampling methods some of them might be better than the others in certain situations. Like you can do ABF, you can do umbrella sampling, you can do umbrella sampling with replica exchange, you can do metadynamics, you can do any of these things. But these are sampling methods. That's not the main thing that you should actually worry about, I believe. What you should worry about is, first, the collective variables. That's really is going to determine what you get out of it. That is in the very first paper that I actually published in my academic work in 2009. I basically had this idea and I tried. This was a polyproline peptide, and I wanted to study the transition between the right-handed and left-handed peptide because it can be found in both. And I came up with something like 5 or 6 different collective variables based on the dihedral angles, based on the just the handedness of the peptide, based on different ideas and combining them, getting different free energy maps, comparing them. And one conclusion that I had was the free energy that you get has a lot to do with the collective variables that you choose. And since then, it's been an important part of any sampling or any computational work that I've done. I try to first spend a lot of time on choosing the right collective variables before going to the next step and making this quantitative. So collective variables and then trying them with simple SMD type simulations is the very first step to figure out whether you are working in the right framework or not for doing your enhanced sampling. Yeah.
So you've used SMD simulations for that? Or TMD?
When I say SMD, or I usually call it non-equilibrium pulling or non-equilibrium driven simulations.
Right.
What I mean is basically pulling on the collective variable.
In that family, have you ever tried this adiabatic biased MD?
No, I haven't tried that.
Okay.
Yeah.
I'd love to know it.
Yeah, I heard it actually from you the other day. And,
Okay. Right, yeah, it's kind of under the radar. It's not very well known.
Yeah.
But I've tried it just on one simple case, and it was a pretty interesting result. So
Yeah.
I'd love to know.
Yeah, definitely I will try it because I heard it from you. I had heard about it before, but I never just paid too much attention to it. So it's definitely something to look into. But I mean, generally, the collective variable is more important than the way that you use that collective variable in your sampling. And some other people have shown this too. I should have probably cited that paper here, but there is a work from D.E. Shaw group. The first author is Alan Pan, who is the one who developed the string method with storms of trajectories. So he shows for a specific system that they had run a very long equilibrium simulation on Anton and had actually observed the transition. It wasn't, of course, the transporter. He had used multiple different methods, like string method with storms of trajectories, SMD, different things, but with different collective variables. And the conclusion of the paper was, it's just a collective variable that determines what pathway you get. Even a string method with storms of trajectory wouldn't be able to get you the right pathway if you don't use the right collective variable for it. And this is coming from the person who invented the string method with storms of trajectories. Quite a bit. Yeah.
But you don't mean that there is only one pathway. It might be multiple pathways to get the same thing. So maybe all of them are okay.
The assumption for slow processes is that usually there is one dominant family of pathways. You can think about the transition tube. Of course, it's not one pathway. It's a tube. It's like a family of pathways that are close to each other, which dominates because when the processes are so slow, if you have two pathways and one of them happens every second and the other one happens every minute, you're talking about very slow processes. Then the one that happens every minute is not really as important as the one that happens every second because, it happens just more often. Sometimes it might be, but the difference is usually, and think about it in terms of the free energy. So if one of the barriers is 8 kcal per mole and the other one is 10 kcal per mole, this 2 kcal per mole difference in the barrier would translate into about, 100 times more likely for the other pathway to be. Yeah.
So maybe one problem that could arise then is there are multiple pathways. Maybe only one of them is genetically relevant, but there is a risk that your simulation could explore one.
The other one.
One that is.
Absolutely, yeah. So there is really no mathematical, I believe, answer to that question. So this is simply something that everything that we find from these simulations need to be tested experimentally. Absolutely, I do not believe in any mathematical way of figuring out whether there is another pathway, unless you just try to explore multiple pathways and compare them and see which one is better. And you may never end up with the right pathway. So this is actually a caveat which exists there. Okay, so this idea, so I'm kind of going over these ideas that I have here in different forms, in different formats, and then talk more specifically in specific problems in the second session. So this is one graph that kind of summarizes the workflow. You start with defining practical collective variables, and the VMD GUI for collective variables would be the best option for this. Then, what I call here optimizing the biasing protocols is you are going to combine these collective variables. You are going to use these collective variables to push the system using non-equilibrium pulling simulations or SMD, and still it requires some optimization, especially if you are going to compare different pathways. For instance, suppose that you have two different collective variables. One opens one side of the protein and the other one closes the other side of the protein. How are you going to combine them? Are you going to combine them just linearly? Are you going to do one first and the second one next? In what order you are going to do this? So one of the things that if you want to start doing some combinatorial search on how to combine them, then it's a good idea to also choose the same, for instance, the right force constant for them, choose the right simulation time for these non-equilibrium simulations that you are going to run, and how these optimizations can be done. One of the ways to do these optimizations is to look at the non-equilibrium work and try to optimize your biasing protocol in general in a way that gives you a non-equilibrium work which is not too large. So the non-equilibrium work is one of the measures that you can use to optimize your biasing protocol before going to things like string method and free energy calculations. And I will show you an example of that how we can look at the non-equilibrium work and choose which collective variable or which combination of collective variables and what force constants we want to use. Yeah.
So because you're using biased forces, right?
Mm-hmm.
So the events what you see in your simulation, how do you know it's not an artifact of?
It could be an artifact, and that's why this is not the end. So this is just the beginning. So we have all these other steps to make sure that at least we have a self-consistent idea about the transition pathway. So the next step would be taking your non-equilibrium trajectory, which is generated using your non-equilibrium pulling protocol or your SMD simulation, and optimize it further. And for optimizing that, for instance, you can use string method or other pathfinding algorithms. And there are other terms here that I will explain, especially when it comes to the iterative flow. Then there is another analysis tool that I've developed called post-cycle string method that comes very handy. So once you have optimized your transition pathway, and I assume you have heard about string method with terms of trajectories in previous talks, the next step would be free energy calculations. And again, for free energy calculations, you can use different methods. You can use ABF, EABF, metadynamics, umbrella sampling with replica exchange. many other methods, which are based on collective variables that you have. And one particular type of free energy calculations that goes very well with the string method with terms of trajectories is the simplest one, the umbrella sampling. Because umbrella sampling, especially if you combine it with replica exchange, and I call it bias exchange umbrella sampling, allows you to take the pathway that you generated with the string method, use the exact same images of the string method, exact same biases of the string method, and just probably lower the force constant, because the force constant used for a string method for restraining is usually high. Just lower the force constant so you can allow for overlap and exchange, and then just run that simulation. So it's very natural. It's a very natural step after doing a string method with terms of trajectories, because it can actually use the exact same setup. It can even use the same number of copies that you used for your string method for each image to run the simulations. And this is in the tutorial and in the scripts that are with the tutorial, you can do that. If you do have if you do have a large supercomputer available to you, that you can use like multiple copies for each image, then you can use the exact same copies for your umbrella sampling with replica exchange simulations as well, and it improves the sampling. It also allows you to do error estimates, because now you have multiple copies of each window, basically, and multiple independent trajectories that you can use for error estimates of your free energy calculations. The next step that is sometimes ignored is to check the outcome. So, of course, you need to do some sort of reweighting to get the free energies. The most common one is VAM, which is not so much relevant here for multi-dimensional spaces. If your collective variable is like a multi-dimensional one, if you have like 32 collective variables in the space that you are doing your free energy calculations, although it's along one single path, but VAM would not be the right method to use there, because it asks you to build a histogram in a 32-dimensional space. But you can use a non-parametric reweighting scheme, such as M-bar and related methods. And I will mention some of those methods later. So that's something people do. But then one other thing that you can do is, or you should do, is to check the other degrees of freedom. So you have a number of collective variables. You have done your free energy calculations. You have made sure that there is overlap. In the replica exchange scheme, another thing that you need to look at is the exchange. So I've seen these papers in the literature. They use replica exchange simulations, bias exchange umbrella sampling, replica exchange umbrella sampling. They don't even report whether there was an exchange between these windows or not. I've seen a paper published in Nature that uses replica exchange umbrella sampling and doesn't say whether there was an exchange or not. So if there is no exchange, what's the point of actually running the umbrella sampling with replica exchange? So this is something you need to check. You can look at other degrees of freedom. One very, very simple thing that can be done is you can simply just do PCA on your trajectories that you get out of your free energy calculations if you have multiple windows. You can do PCA and then look at the sampling. See if your sampling is continuous or not. It's happened to me several times. So I've done the sampling after doing PCA, looking at the PC1, PC2, I see two separate clusters of samples. Because somehow my collective variables do not cover everything, and this is less common here when you use a multi-dimensional collective variable space, but especially if you are using a one-dimensional collective variable or a two-dimensional collective variable. There is no particular reason that other degrees of freedom are going to be sampled continuously. So if you do PCA analysis and plot your data, all your data coming out of all of your simulations into the PC1, PC2, PC2, PC3, PC1, PC4, if you plot them in different spaces and look at them and don't see a continuous sampling, then your results are all questionable. And it's very, very simple to do. It doesn't, if you do PCA based on the C alphas, that's very simple, very fast, and it tells you something, but still it may not tell you about the sidechains. So especially the important sidechains, you need to make sure that they have been sampled continuously. So if you plot certain quantities, like dihedral angle, the chi1 dihedral angle of an important sidechain that is involved in binding, plot that along the windows. So for every window, just measure the value of the dihedral angle and plot those values along. The x-axis would be your windows, the y-axis would be the dihedral angle coming out of different samples. Or Soul bridge distances, local things. So you should, especially if your collective variables are based on global things, you should make sure that your local measures are all well behaved. You have continuous sampling. If you don't have continuous sampling, if you have two windows, in one of them your sidechain is up, in the other one your sidechain is down all the time, you may have exchange between them. You may have overlap between them in the collective variable space. But eventually that is going to be problematic for your calculations. The replica exchange part is going to be helpful to recover some of that, because when you have exchange, basically it allows for different different conformations of this sidechain to be put in different windows and sampled. But eventually, if there is any discontinuity in any space, in any along any collective variable, then the assumptions of your reweighting schemes are not going to be valid anymore, and it's problematic. So, well, why do we need to do all of this? Well, we do need to do these because if we don't, our results could be wrong. It's very simple. So you may end up with free energies that are simply just wrong. You may end up with a barrier which is very low. You may end up with a free energy difference between your end states which is not correct, which does not agree with the experimental free energy differences. And that's why we do need to do this. And if we continue not doing these things and publish our results and generate these numbers that don't make sense, then we're going to lose, as the molecular dynamics community, we're going to lose our reputation. People are not going to trust us. And this is actually something that is happening. I see these practices in a lot of papers that are being published. They do not report any testing of any of the results, validating statistically what they have, and what they publish doesn't really make much sense. Like you see these free energy profiles with very low barriers for, say, a transporter. And I usually look at the transporter papers because that's the area that I'm more familiar with. And I see these barriers like 2 kcal per mole, 3 kcal per mole barriers for the transition of a transporter. It doesn't make any sense because transporters are very slow. How come it's 2 kcal per mole, 3 kcal per mole, but the transition occurs only once every 200 milliseconds? So they just do not agree with the kinetic studies. And the reason should be actually looked into by just self-consistency assessments. So if you get the wrong answer, of course you can always blame the force field, but in my opinion, most of the times if you don't get the right answer, it's because of the sampling inefficiency. Okay, so this is my first part of the talk this morning, and I will continue with more specific examples. So far I kind of focused more on the general ideas, and now we're going to go deeper into some examples and some of these methods that have been used for those examples, and hopefully that would make it more sensible. Thank you. Yeah.
So the idea behind enhanced sampling or these methods is to get all the sampling in a reasonable amount of time. So, for example, if you have a process that takes a microsecond or a millisecond, you want to still be able to cover that process in, let's say, 100 nanoseconds or 500 nanoseconds. So, do you have a ballpark number for different methods? Like, how long should the simulations be?
Well, basically the idea is you want to limit your sampling to the interesting areas of the phase space, which is a very large space, and this is very system dependent. So, and it's also very dependent on collective variables. So my idea is if you choose the right collective variables, it's going to be a lot cheaper to do the free energy calculations at the end because they converge faster and they give you actually results that do pass all those checks. If you don't use the right collective variables, it may never convert, or it may never actually pass those tests. So I can give you examples. The particular example that I'm going to show mostly used about 2 microseconds of simulations altogether, in aggregate 2 microsecond simulation for reconstructing the entire transport cycle of a transporter, which doesn't just involve one conformational change. It involves binding of a substrate, undergoing conformational changes, unbinding of the substrate, undergoing another conformational changes to go back to the initial stage. So to reconstruct that entire process, we used something like 13 different umbrella sampling and string method and different methods combined in this iterative manner, and the whole thing was about 2 microseconds. So it's still expensive. Okay, thank you.